This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV, this is TWiM, This Week in Microbiology, a podcast that explores unseen life on Earth. Hi, everybody. I'm Vincent Racaniello, and joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
Welcome back. We missed you.
Yeah, I've been AWOL for a while.
And happy birthday.
Oh, thank you.
On Elio's blog yesterday, a little celebration of his birthday, and we join in as well. Terrific. And welcome back, of course. We love having you here and your wisdom and all that.
Oh, boy.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, and I want to wish Elio many happy returns on the day.
Oh, thank you. Thank you.
It's great. Many more. Many
I don't
more.
really know what that expression means, but it just sounded great.
I don't know if returns are.
Noble.
Noble.
Also joining us from Charleston, South Carolina, Michael Schmidt.
And I'd like to echo my happy birthdays to Elio as well.
Okay.
We actually had cupcakes in your honor yesterday.
Oh, really?
We went out to lunch and had cupcakes. Now, it was a colleague's birthday as well, but she is younger. But
everybody's younger.
Almost. There are some people who are older. You know, I saw an article about a 100-year-old woman the other day who still runs.
Yeah, I saw that.
Did you see that?
Yeah, amazing.
Wow. Must be special genes, I guess.
on the original knees?
She's
she
a
has
world
new
champion
knees?
in her age group.
Yeah, apparently. I don't know what's artificial in there or not, but it's pretty impressive either way.
Yeah.
Also, you, Elio, you have a new book coming out to celebrate the 10th anniversary of Small Things Considered. That's very nice.
That's right. Yeah, it's called In the Company of Microbes, which goes with the title. The title comes from my having written a book once called In the Company of Mushrooms.
Mm-hmm.
So now I got the franchise of In the Company of.
That's great.
Yeah, it should be fun. It's a bunch of pieces that people have written for the blog, and I chose what I thought would be fun for a more general public, let's say.
I think I have one in there, right?
Yes, I think you do. So does Michele, and yeah.
Oh, that's great. Looking forward to that. When is that going to be coming out?
Well, it's advertised in Amazon. It says May 1st.
Great. That's pretty soon.
Very soon.
And of course, Microbe is also coming out. I think that's going to be at the ASM Microbe meeting, right?
Yeah, the textbook.
I'm looking
Microbe, the textbook, as opposed to Microbe, the meeting.
Wow. Got a
And
brand.
Microbe, the magazine.
Wow, what a brand.
It's interesting. You know, the word microbe is taken over from microorganism.
Mm-hmm.
Really taken over. A very sudden shift. And we had something to do with it. And I'm very glad because it sounds nicer than microorganism. You know,
I like microbe. I think it's a wonderful word.
Yeah.
And microorganism is a little bit sciencey.
That's right. So stodgy.
But yeah, microbe is great.
Good.
Well, this episode of TWiM is brought to you by the American Society for Microbiology. They want you to know about their second Auger Art Contest. This is a contest where you take a petri dish and streak out or drip bacteria on it in an artistic way. And last year we did this. We had wonderful, wonderful pieces submitted. And in fact, I was at a meeting recently, and someone came up to me and she said, I won the Auger Art Contest last year. So I was very happy to meet her. And then you take a picture of your creation and send it in. And you can find all the details at bit.ly/AugerArt2. The deadline is coming up May 6th, and the winners and runners-up will be showcased in a special gallery at the ASM Microbe 2016 meeting, which will be in Boston in June. And all winners will also get a free ASM book of their choosing. You can choose Microbe or In the Company of Microbes, or you could choose Principles of Virology 2.
Oh, God.
Hey.
Are you shamelessly promoting your book, Vincent?
Well, they're not going to sell it, so it's not like any royalty is forthcoming, right? They're giving it away. But yes, I put it at the end. I know that Microbe and In the Company of is first. Anyway, bit.ly/AugerArt2. We thank ASM for their support of TWiM. So today we have a snippet and a paper, and we welcome Elio back. And Elio has a nice snippet for us. Tell us about that.
Okay. The snippet is based on a paper by, the paper is entitled Microbiological Characterization of Post-Eruption Snowblower Vents at the Axial Seamount Juan de Fuca Ridge. Now, that's a lot of words that are probably not terribly intelligible. The authors are Julie Meyer, Nancy Ackerman, Giora Proskurovsky, Julie Huber. They are from the Woods Hole Marine Biological Laboratory, as well as from the School of Oceanography at the University of Washington in Seattle. Okay, now to explain what this is all about, it's mainly about the bottom of the sea. Not so much there's obviously there's microbiology in here, but I think it's interesting enough to tell you about what it is that we're talking about. Let me start out. In 1997, a great event happened in our globe, namely the discovery of hydrothermal vents. 1977. These are, as I think most listeners know, are vents in certain ridges where there are the tectonic plates come together, and this is where there's a constant eruption of some black stuff. The black stuff is outpouring of dark particles. This is why they're called the black smokers. And they're black because of tiny mineral particles. Now we're talking about, and that a tremendous amount of work has been done about this, including the finding of a lot of life there. The giant tube worms are the best known. They're sort of meter-high worms, which are full of bacteria which oxidize sulfide and give the source of the energy for the worms. Anyhow, lots of crabs and mussels and other animals and lots of microbes are there. As opposed and contrasting with the hydrothermal vents, there are also things called snowblower vents. They were discovered in 1991, and they are low temperature, ephemeral phenomena, it said. That is, phenomena that come and go. The temperature is not 400 degrees of the hydrothermal vents, but 12 to 17 degrees centigrade. Now, what characterizes them is two things. One is a copious outpouring of white flocculent material, often in recent erupted lava area. These look like a snow globe effect. The clouds of rising fluid carry white fluffy particles reminiscent of snow. The material, the snow, unlike in the high temperature black smokers, where the suspended material is minerals, this is actually biological in origin and is a byproduct of the microbial bloom that happens in the subsurface hydrothermal system. In other words, there's lots of life down below. The surface of the sea, the subsurface of the sea is just full of life, and this is the effect of having life down below the surface, the bottom surface of the sea. The volcanic eruptions that happen once in a while, what the authors say, invigorate the subsea floor biosphere and create these blooms. Now, the place to study this is a place called, as in the title, the Axial Seamount. That's the name of the place. Okay, you gotta live with it. It's a seamount, meaning a high-rise place on the bottom of the sea, which has a submarine volcano, and it's located in the Juan de Fuca Ridge, which is an important ridge, 300 miles west of the coast of Oregon. It's the most active volcanic site in the North Pacific, and it's been studied in good part because the National Oceanic and Atmospheric Administration was granted access by the Navy to a chain of submerged hydrophones, which had been used by the Navy to detect submarines in the Cold War. So this is neat because this Cold War relic, they're still there. These hydrophones can be used for studying interesting geological and biological material. But however, the axial seamount was detected of all things by satellite altimetry in the 1970s. It was explored by several different, a whole raft, in fact, of submersibles like Alvin and others. They also dropped a large package of sensor on the seamount and established a thing called the New Millennium Observatory on the flanks of the seamount. So it's a place that's been studied very, very intensively, and it followed a submarine eruption of the volcano in 1998, and it's the first time that a submarine eruption has been detected and followed right then and there. So this is an interesting place. So keep in mind the term snowblowers. Okay, that's going to be part of our understanding of the geosphere from now on. Now, around the snowblowers is lots of life, including tube worms of the same kind that you find in the high temperature vents. And they form big, huge mats up to 6 square meters. Sounds like a lot. And you find also bacterial mats, smaller worm tubes, limpets, and other life. Normally, these snowblowers are covered up by lava. They're inside, below the lava, below the surface. However, a vent was created when one of the submersibles accidentally punched a hole in the roof. The hollow lobe had been an incubator, essentially, a warm water filled with microbial floc, which came out of the hole when this was. Okay, so now the microbiology. All this is microbial stuff, right? Microbial origin, microbial nature of these white flocs has been known for some time. And for a while, some people made a very interesting guess. They guessed that these were epsilon proteobacteria of the genus Archobacter, which I don't know much about. And the reason they thought so was interesting. An isolate of this genus from salt marshes made very similar white flocculent material in the lab. And this was the white stuff was bacteria and excreted elemental sulfur. So that sounded like, gee, if we can do that in the lab, we can do it under the sea. However, Archobacter is only found in a very small fraction of all samples, so this was a wrong guess. Anyhow, now the authors point to something new, namely, besides the white floc producers, there's also in the near the snowblower vents orange flocculent material that coats the seafloor in surrounding the sites where multiple seafloor eruptions took place. Now, the orange floc is different from the white floc in several ways, and it's colonized by a different subset of microbes. These are gamma proteobacteria instead of epsilon proteobacteria. And the reason that this material is brown is because of iron oxidation. And sure enough, you find iron oxidizing bacteria in this stuff. So there are two areas, some which give you white flocs and some which are coating the seafloor, which are brown in color, or orange, really. So they studied both. And I more or less told you already the grand conclusion, which is that two different populations, and they all fix carbon through rubisco, the enzyme that does most, if not all of the most of the carbon fixation in the world, and they oxidize hydrogen sulfide, therefore producing elemental sulfur. Okay, now. Some enrichment cultures that they obtained can produce the white flocculent material in the laboratory as well. So that's pretty much the story. They go into further detail because in the white flocculent, the organisms that are most abundant are, as I say, epsilons. They are mainly sulforovum, and sulforimonas, which are obviously with the word sulfur in front of them, are sulfide oxidizing bacteria. And they are likely residents of the shallower subsurface. There's also some methanobacteria, methanosacinalis, which probably are being flushed from even deeper subsurface layers. So they got from this material. They sequenced it. They got over 3 million. They call perfectly overlapping reads, both from bacteria and archaea. There are not many bacteria, by the way. There are only about 1% bacteria. Archaea. They're mainly bacteria. So the story here is that they found in the epsilon bacteria the sulfide oxidation gene SOX-B, which was found throughout a lot of these organisms. There are 4 phylotypes with a 90% amino acid level similarity from 71 clones. Anyhow, none of them are shared between the white and the orange flock samples. These are all white. And as I said, Sulforovum is the dominant genus. Okay, so that's the orange flocks had different kind of SOX-B genes. There were only 85 similar to the ones in the white flocks. Okay, that's pretty much the paper. So it's interesting, I think, mainly for the awareness that it brings up of the snowblower areas. But it also says that despite the difference between snowblower vents and what they call the more stable diffuse flow vents, in other words, the vents which are not, they don't make chimneys and they don't make these structures. But anyhow, the dominant organisms in snowblower fluids, as well as in the white flock, are similar to diffuse flow. So there's elsewhere in the ocean we find the same organisms. So there's a lot of life down there, and it probably contributes in a significant way to the biogeochemistry of the planet.
Do we think that these snowblower vents are throughout the ocean floor or just in certain places?
No, they're probably found mainly on the mid-ocean ridges, the places where the tectonic plates come together.
And that's where the volcanic activity is mainly found?
Yeah, they depend on volcanic activity.
I see.
And I think it bears emphasizing that all of the deductions, all of the conclusions they're making about the metabolism of these microbes are deduced from genome sequences. Because, of course, they don't have live cultures.
Welcome to the new world.
Right. They don't have live cultures from these
They
clouds,
have some
right?
cultures, yeah, but not much.
But they do have spectacular images. There are some beautiful scanning electron micrographs and some other epifluorescent images that are really quite attractive. We're going to put them,
Vincent, you're going to put them in the show notes, aren't you?
Yep.
I urge the listeners who are interested in going and having a look. There's some movies which are totally spectacular.
What a cool way to get a glimpse of what the microbial communities are in these deep, deep vents.
Yeah, it's amazing.
It's kind of nature's experiment. They're spewing them up so we can capture and have a look.
What I find amazing is that we didn't know about these until pretty recently, right?
That's right.
It's just right here on Earth. There's so many things probably still to be discovered. These are great galleries of images, and there's a video as well. We'll put links all up to those. This is very cool.
We should talk one of these days about the Banfield new Tree of Life, which
sure
you've probably seen, and which indicates there's much more diversity than we thought.
Oh, yeah.
Infinitely more.
Well, I heard a talk the other day by Eddie Holmes, who is a evolutionary virologist, and he is sequencing insects, and he finds so many new viruses. The phylogenetic tree goes up by an order of magnitude when you put it's just amazing what's out there that we don't know about. It's just incredible. Well,
That's the power of sequencing, isn't it?
It sure is. And as we were saying in the pre-show, this the desire to Michael, you said the desire to look at the microbiome pushed deep sequencing forward, right?
Yeah, it did, because it dropped the price so phenomenally. I think it's dropped an order of magnitude in costs in the last 10 years.
I just saw
much more than that. Much more than that.
And the computational power
too.
Has increased.
Yep. Yeah, that has gone along with it, because you need that to be able to analyze the data, right?
To interpret it, right?
The days where I could look at the polio genome and do things by hand, right?
Are long gone.
I mean, when I did the genome in 1980, I did everything by hand. I used to mark it up. So, look, here's this protein, here's that protein. You can't do that anymore.
I can remember sitting at the light box, type in with one hand, GAAT.
Absolutely.
We're now telling everyone how old we all are.
You had the gel on the light box, and you were typing the sequence as you read it, right?
Right.
Uh-huh.
I wonder what the error rate of that was.
Well, that's why you did it over and over again.
We didn't do too badly.
Well, and you had good coverage from.
You had good
directions.
coverage.
Yeah.
Yeah, we didn't do too badly for all that. Anyway, thank you, Elio. Very interesting. All right, I have a paper for you about Mycoplasma. Now, this is the second time we've talked about Mycoplasma within the last few episodes. And just to refresh your memory, we talked about a synthetic Mycoplasma genome in TWiM 125. So the normal genome of this particular Mycoplasma was 1 million base pairs, or is 1 million base pairs. And Craig Venter's group made a synthetic Mycoplasma genome, about half the size, 531 kilobase pairs. And we talked about that in TWiM 125. But today, we're going to do some old-fashioned biochemistry on these very small bacteria. They have the smallest genome of any autonomously replicating cells. Is that fair to say, Michele and Michael and Elio?
It sure is.
Yeah.
Okay. They have no cell wall, limited metabolic pathways. However, they are pathogens. They can infect humans. They can infect other animals. And some of you may know the human pathogens, Mycoplasma pneumonia, Mycoplasma genitalium, which cause, as you might guess, respiratory tract and urinary tract infections. Now, these are infections associated with a lot of morbidity, a lot of disease, but not a lot of death, low mortality in general. Now, in animals...
Let me say two things about Mycoplasma.
You bet.
One of them is that even though they don't have a cell wall, they're almost ubiquitous. Therefore, they're pretty tough. Mm-hmm. So it doesn't take a cell... Normally, you think of the cell wall as being the protective envelope of most of all bacteria except these, but they manage. They manage very well. But they have tough membranes, probably. They have steroids in the membranes, and maybe that toughens them up.
Mm-hmm.
And anyone who's done tissue culture will appreciate, because a lot of folks who don't practice good aseptic technique, their tissue culture is often contaminated with one of their own very Mycoplasma that they have brought into their cell culture line.
The other thing is that small genome and small size don't necessarily mean lack of sophistication. These guys have a movement on surfaces that's totally unique. They look like schmoes crawling along, but they do it in a very specialized way, and they have a very complicated apparatus at one end, so that it's very surprising that something with these few genes can do something as sophisticated as that.
Yep.
Misleading, isn't it?
And even though they don't cause, dreadful disease like Zika virus, they cause more what we call a walking pneumonia, which is another successful strategy. So if you're not home in bed and you're out walking around and sneezing, it promotes its transmission in the population. So these are highly successful pathogens.
Right. Yes, I did not mean to imply, of course, that they were unsophisticated. But Venter showed that you can cut the genome in half. Elio, too bad you missed that episode. They can cut the genome in half, and the doubling goes to like 20 minutes from much longer. It's amazing.
It is.
Anyway, in animals, Mycoplasma infections have a lot of economic impact. For example, in cows, there's a disease called bovine pleuropneumonia, caused by Mycoplasma mycosis, a big problem in Africa, up to 50% mortality in herds that don't have any immunity. There are also diseases of goats, sheep, pigs, chickens, turkeys. So you can understand
also,
why.
There are somewhat distantly related spiroplasmas, which are very important plant pathogens.
Yes, they also infect plants. And that's an interesting thing we'll get to towards the end of this. Now, in general, we don't understand a lot about the pathogenesis of Mycoplasma infections, and they don't appear to have secreted effectors, as many bacterial pathogens do. They have a limited number of toxins. So what else is there? Many infections are chronic, right? They go on for long periods, as Michele said, and that suggests there are some mechanisms in place to evade immunity. Otherwise, they should be cleared. And some of the things we know include antigenic variation of the surface proteins of Mycoplasma. And it's also been suggested that they can in some way target antibodies of the host, what we call immunoglobulins. There's some evidence that IgA can be cleaved, but the protease has not been identified. And there's also a protein called protein M that's been identified that binds multiple immunoglobulins. And they're wondering what's going on there. And that's actually the subject of this paper. And the name is MIP, is a mycoplasma system that captures and cleaves immunoglobulin G. And the first author is Jonathan Arfi, and the last author is Pascal Siron-Punier. And this is a group of authors from France, the University of Bordeaux. Wonderful. They must have nice wine at dinner there.
At faculty meeting, I'm sure.
The University of Grenoble in Grenoble, France, where I think the Olympics were once, right? Many years ago.
Correct.
What else do we have here?
And the University of Bern in Bern, Switzerland, also Kenya.
And also our friends at the Craig Venter Institute in Rockville, Maryland, must have had some help with sequence analysis there.
And somebody from the Livestock Research Institute in Nairobi, Kenya as well.
There you go.
Boy, is this ever international.
Sure is. And this is published in PNAS. So they work on Mycoplasma mycosis capri, which is associated with respiratory diseases in small ruminants. And they had been looking at the genome, and they found an atypically organized cluster of genes where they had two genes repeated 4 times. And what they encode appear to be proteins that would be, one of them would be a predicted serine protease, and another protein whose function they didn't know. They have transmembrane domains, and so they say they're probably at the cell surface. And they show that all these genes are actually expressed in the mycoplasma during growth in medium in the laboratory. Now, the protein that doesn't have any homology has weak homology to protein M, which is the binder, the immunoglobulin binder that's been previously described. So what they did is they took the gene for this protein, they produced the protein in E. coli, and in fact they show it binds something in goat serum, and eventually after some mass spectrometry they identify 2 proteins as the heavy and light chains of immunoglobulins. Everyone knows an immunoglobulin or an antibody molecule is a Y-shaped molecule. It has 2 heavy and 2 light chains, and the antigen binding sites are up at the tip of the Y molecule. So they show that this protein from Mycoplasma binds immunoglobulin from sheep, cow, horse, mouse, and human sera, but not from rabbits. So therefore, it's an M protein that's binding immunoglobulin. So they call it MIB, and that, I guess, is M immunoglobulin binder. I remember it as Men in Black. That's a movie I really liked. Okay, then the other protein is next. This is this putative serine protease. Again, they produce the protein in E. coli, and when they incubate the protein with IgG, the heavy chain gets cleaved. But the protein by itself doesn't do this unless MIB is present. So you have to have both MIB and this other protein, which they call MIP, or protease, together. And then the 2 of them, and presumably the protease cleaves IgG. And this works with IgG from sheep, cow, horse, mouse, human. Now, the interesting thing is that the activity of this protease, the serine protease means, so proteases have active sites, and there's typically a catalytic triad, and one of them is serine, and that's characteristic of serine proteases. There are inhibitors, chemical inhibitors, which basically bind the serine and inhibit the enzyme, and they use several serine protease inhibitors, but they didn't inhibit the activity of this particular protease. But when they change the serine in the active site to alanine, that kills the activity of the protease. So they say this is a serine protease, but there's something different about it. And what that is, we'll get to later. So where does the protease cleave the immunoglobulin? So they go through a series of neat experiments. They show that cleavage gives, of the heavy chain that is, now remember it's cleaving the heavy chain, gives a 44 kilodalton fragment, and they eventually sequence the N-terminus and find that the cleavage occurs between the VH and the CH3 domains. So a heavy chain has 4 independent IG-like domains. They're called VH, CH3, CH2, and CH1, going from the top to the N-terminus to the C-terminus of the protein. So this is cleaving precisely between the two domains. And the cleavage site, the N-terminus, the new N-terminus that's produced as the sequence VSS-AST. And they found this sequence in sheep, cow, horse, human, and mouse IgG. And of course, those are all cleaved, so that makes perfect sense. Okay, so this protease is cleaving IgG heavy chain, and it's cleaving between the VH and the CH3 domains. And by the way, these parts of the molecule they contain the antigen combining site. They do some very nice binding studies, and they find that MIB or MIP separately cannot bind immunoglobulins. So if you add MIB to IG, it doesn't bind. If you add MIP to IG, it doesn't bind. But if you mix them together first, then they both bind. And they use chromatography. Can you imagine? They run columns to find the size of the protein and, whether it's a monomer or a dimer. And they basically find that when you add MIB, the binding protein, to IgG, you get 2 bound, 1 for each FAB domain. So if you think about an antibody being a Y molecule, the 2 upper parts of the Y, those are called FAB fragments. And the bottom is the FC fragment, which binds complement and does other things. So these MIBs are binding each FAB fragment. And then the MIP binds each of those MIBs in turn. So you get 2 of each bound to each arm of the antibody Y, if you will. And they say this is very stable.
Shown beautifully in their schematic, figure 3A.
Right, on the nice
architecture
picture of the antibody.
And
they actually run gels here. Can you imagine? Protein gels. They do reducing and non-reducing gels. Hardly anyone does this anymore, and they're so beautiful.
They're gorgeous.
They tell you a lot of information. In the same way, people don't do northern blots. They do PCR and sequencing. But boy, this is just great. This is one of the reasons I like this paper, Classical Biochemistry. So that is pretty much most of the data. What they think is that the binding of, so remember the serine protease inhibitors did not inhibit MIP. So what they think happens is MIB binds to the antibody first, and then MIP binds to MIB, and that binding causes a conformational change in the protease, and that exposes the catalytic serine. And so that
I love that drawing, by the way.
Isn't that nice? It's a very nice figure showing the exposure of the catalytic site, which is normally hidden. So that would explain why the serine protease inhibitors don't work. And also why you need MIB to bind MIP to bind the antibody and get cleavage. Without MIB, so it's a 2-component system, really. You need MIB to bind the antibody and then to activate the protease. So they searched through all the sequences they could, and they found MIB and MIP homologs in most animal pathogenic mycoplasmas. So they suggest this may be important for defense. But interestingly, they were not present in mycoplasmas that infect plants.
I loved that result.
Isn't that great?
Because there's no immunoglobulin?
Yeah, I guess.
Yeah.
It would be
think?
It would be no selection. And that may be telling us that there's been pressure to maintain these in the genomes of these other mycoplasmas, right?
Well, that's the hallmark of the mycoplasma genome. It gets rid of things it no longer needs, because that's how it's able to maintain itself in the host, because it got rid of its cell wall production system, which is huge. And I think the plant story, fits nicely with the mycoplasma being very, very sophisticated, as Elio said in the beginning.
Yep. Now, interestingly, MIB and MIP homologs are not in the 2 human pathogenic mycoplasmas. Right now, we do make antibodies, obviously. So they say, well, human mycoplasmas have an M protein, an immunoglobulin binding protein. It's not MIB, but it looks related to it. And they also see there are some proteins related to MIP. So that has to be studied, but maybe there's something there, and it's just a different one, which would be interesting, right? So humans have one kind of MIB-MIP, and the other mycoplasmas have a different one, perhaps. The final thing is that the cleavage site on the antibody by MIP is unusual, and they say all proteases in bacteria that cleave immunoglobulins, they cleave between the FAB and the FC portion. So that's called the hinge. So if you think of a Y, You have the two arms, and where the two arms come together, that's the hinge. And if you cleave there, you separate the FC from the FAB. And they say this is what most known bacterial immunoglobulin proteases do.
IgA proteases.
IgA proteases. Only IgA?
Right.
So that would remove, when you do that, you would remove the complement binding protein portion, the FC, from the antibody from the FAB. And of course, complement attaching to antibody is an important part of the activity of antibodies when they're bound to an antigen. Now, so what is the Mycoplasma protease doing? It's cleaving in a different place between VH and CH domains, and that removes 3 of the 6 regions involved in recognizing antigen. So there are 3 in the VH and 3 in the CH region of the variable domain, the upper parts of the Y of the antibody. These are called complementarity determining regions. So, cleaving the antibody is most likely going to reduce its ability to bind antigen. So it could be an immune evasion mechanism. So basically, clearly the next step we have to do here is to look in animals infected with these mycoplasma and see if cleavage of the antibody plays a role in immune defense. I guess you could put in mycoplasma lacking an active enzyme and so forth and see what it does to the pathogenesis. That's about it.
You're not going to comment on their elegant stoichiometry where they explain to you how the math adds up?
No, you can do that if you'd like. I decided that I didn't want to.
You didn't want to explain the stoichiometry.
I just told everyone the result.
All right,
and I think that's okay. If you are interested, you can go back. But I have to make a decision, and I made an executive decision that it wasn't. I didn't want to do it.
I really loved the parallel with the IgA proteases that a number of human mucosal pathogens make. Mm-hmm. So strep pneumoniae, H. flu, Neisseria, both meningococcus and gonococcus, they each make enzymes that cleave in the hinge regions of IgA proteases, presumably because IgA is abundant on those mucosal surfaces, so they have to counteract that with these proteases. Right. So I love that this was a parallel story, but now they're targeting IgG.
Mm-hmm. There's even IgG protease in Pseudomonas.
Mm-hmm. Mm. Now
I just looked it up. I didn't know that. Pseudomonas in cystic fibrosis has an IgG proteolytic activity.
So all the mycoplasma are classified in a class called mollicutes, and only mollicutes have this MIP system, right? So it's interesting that it has evolved in this group of bacteria, but nowhere else as far as we know. But again, that could change as well.
And I'm not sure you said it, but they have evidence that it's transmitted by horizontal gene transfer. Mm-hmm. So it's a cassette that is spreading among that genus, which is kind of cool.
Among the mycoplasma, right? Yeah.
Yeah.
That's right.
I was able to correspond with Jonathan Arfi, so I'd like to tell you a little more about the first author. He's a French citizen, son of a biology teacher and an oceanographer, and he is currently what is called maitre d' conference, which is the equivalent of an assistant professor. So he does both research and teaching. He said he was interested in science at a young age, probably thanks to his parents. He was an undergrad at University of Marseille, where he studied biochemistry, and then he got a microbiology and biotechnology master's degree before embarking on a PhD study in the field of fungal biotechnology. So he was studying mangrove ecosystem, looking for new fungal species that have unusual and interesting enzymes. For his postdoc, then, he joined a lab at the Weizmann Institute, and that's where he got his start in taking what he knew about molecular biology and biochemistry and applying it to synthetic biology, which we've been talking about recently here on TWiM. So he's been with Pascal's group studying the Mycoplasma for a couple of years. He wants to credit Pascal with having done the bioinformatics to recognize this interesting gene pair. And then the two of them together designed this project to understand the function. And Jonathan described the really most exciting day in the lab was when he did a really elegant biochemical experiment that you mentioned, Vincent, where he simply combined the substrate, the IgG, with the two proteins, the MIP and the MIB, incubated them for just 15 minutes, ran it on a gel, and saw a size shift. Mm-hmm. So that's how he knew that he did indeed have a protease and knew that this was a really beautiful project and cool biology. So they're still working on this project. A current goal is to determine the 3D structure of the protein complex as a key to understanding its mechanism. They also were curious about why some strains have multiple copies of this tandem gene pair and what impact that has on virulence. And one of the strategies that they'll use is they're making deletion mutants so they can do the animal model experiments that you suggested, Vincent. He also is involved in two other cool projects that are more related to what we discussed on TWiM 125, which is how can we exploit this tiny, efficient bacterium for good. So they want to design a platform using Mycoplasma to develop a variety of vaccines. So they call this their MycoSyn-Vac program. And then they have another program called their mini-cell program where they want to use a minimalist Mycoplasma cell as a bacterial drug delivery system. So in both cases, they're envisioning, much like Venter's group, using Mycoplasma, engineering a small, efficient one as a chassis, which, by the way, is a French word for frame, that they can then use to add different drugs of choice or different antigens of choice for a vaccine. So those are the many things that he's involved with.
Nice. Lovely. I noticed also that this paper, it's in PNAS, it was edited by Roy Curtis III, who apparently now is at the University of Florida in Gainesville.
I didn't know that.
I didn't either. And I looked him up, Elio. He's younger than you are.
I know.
I didn't know. I thought I thought he'd be older.
He's got a bigger beard than I do.
Yeah, he's a huge white beard. He looks a little like Santa, so I thought he was older, but he's a young guy. He's a spring chicken. Thank you, Michele.
My pleasure.
All right. This episode of TWiM is also sponsored by the ASM Grant Writing Course. Grad students, postdocs, early career scientists. If you want to learn how to write a grant or write a better grant, apply for the ASM Grant Writing Course. You'll get personalized feedback on your grant proposal, hands-on experience in the grant writing enterprise during an intensive 2.5-day course at ASM headquarters in Washington, D.C., August 12th through the 14th. You can experience the full heat and humidity of Washington in the summer. But of course, you'll be inside. You'll be inside all day working on your grant proposals. So you have to apply to this. The deadline is June 30th. You can go to bit.ly/GWC16 for more information. We thank ASM for their support of TWiM. Elio, do you remember a while ago the paper you did on pack hunting by a soil amoeba on nematodes?
Yeah.
So one of the authors wrote,
cool.
Stefan wrote, just discovered this nice episode. Well made and nicely discussed comment on our pack hunting paper. Quick reply to the question, what would happen if you add one single amoeba? It would multiply and a clonal pack would do the same thing as observed. I think that question came up during the episode. They were actually grown from clonal cultures. But let's say if only one amoeba was existing before trapping the nematode, no idea if that would be able to kill it alone. Would be a nice side study. Very interesting hypotheses you came up with. We should study some of it.
Glad to be of help.
He sent a very big smiley and said, keep on with this great work. Thank you, Stefan.
Nice.
Anthony sent us two interesting abstracts. The first, branchial nitrogen cycle symbionts can remove ammonia in fish gills. All right, so in fish, most nitrogenous waste is excreted via the gills as ammonia. And they say there are microorganisms there that remove this. And Anthony asks, I wonder if the relationship is really commensal. Without the bacteria in the gills, wouldn't the ammonia still be carried away from the fish? So they call it a commensal relationship. What do you think, Elio? Is this really commensal, or would the fish have a problem without the bacteria on the gills?
Well, I don't know. I doubt it. I mean, it doesn't. After all, the water would carry away the ammonia.
Would seem so. Yeah, maybe
it's an advantage of being aquatic.
Yeah,
but so I think it's mainly in. The good stuff is mainly in for the bacteria, not so much for the fish.
That's right. So that's not a parasitic relationship because they're not harming the fish.
It's mutualism, or no, it's not mutualism.
No, I don't know. There's so many words, and I think they're okay. Never mind.
It's one-way mutualism. I don't know what that's called. He also sent an article, which this is up your alley, Michael, on copper pajamas. A young lady who was in the hospital with an infection, and she was given copper pajamas, bedding, socks, and a hospital gown, and she got better. Now, of course, this is a study of N=1, but are people doing this, Michael?
Yes. The Israelis, there was a clinical trial conducted in Israel. It was funded with funds from Cupron, which is the manufacturer of the fabric. And they published a clinical trial reduction of healthcare-associated infections in a long-term care brain injury ward by replacing regular linens with biocidal copper oxide impregnated linens. And they saw a clinical effect. And that paper was published in 2014, and it shows that if you reduce burden, you're going to reduce infections. It's intuitive in that you remove bacteria and you limit their introduction into the at-risk patient, and you see a better outcome. And I'll put a link. It's in the public domain, this particular clinical trial, and I'll put a link in the show notes for Ray to post.
Well, especially for MRSA, which lives on skin.
Right. And that's effectively what she had.
Yeah.
Yep. Pajamas. But ideally, you'd give it to people as a preventative, right? If they had a surgery coming up and they were going to be in the hospital a long time.
That would make sense.
Put them in copper at the outset.
I want to know, how stiff are those pajamas?
Well, the pajamas actually smell better, too, because they don't take on odors, because the bacteria don't impregnate themselves into the fabric, so they actually smell fresher.
I would think this is very thin copper wires in with a cloth, right?
Yeah,
Right.
It's not all copper. Is it all copper?
No, it's 60% copper, 20% cotton, and 20% bamboo.
Which gives it that silk feeling.
Do you have a pair, Michael?
If you felt bamboo fabrics, I mean, some of the higher end hotels these days are advertising that they actually have bamboo sheets, which are these very, very soft fabrics. And living in a state with textiles, there's a couple of companies standing up that are making bamboo fabrics that are sort of at the high end because it's, again, a renewable. It's not using oil and all of these other things. And, it's completely, natural.
Michael, do you have a pair of copper pajamas?
No, I do have socks, though.
Really?
I do have copper socks.
Nice.
I have bamboo pajamas, and they're very comfortable.
Really? What percent is it? Like 20% bamboo?
I don't know. I'm not wearing them at the moment, but I'll go home and check.
Okay.
But Michele, they do have that texture of almost silk-like, don't they?
It's lovely. Yeah. It has the texture of cotton, but yet it's got that real soft feel.
Well, Gemma had this young lady in this article had a open wound, which was MRSA positive. So the idea, and it apparently after a few days, cultures came back negative and the wound healed. So,
but presumably she was also on antibiotics.
Yep.
Oh yes.
Yes. So cause and effect is a little tricky here.
Very tricky. And also she could have gotten better on her own as well. So you need to do a bigger trial, right?
Yeah. And the study that was published in the paper was in the International Journal of Infectious Diseases. They had controls and everything. So you can take a look.
Interesting. All right. So the last few episodes of TWiM, we've had a little discussion about the use of the word guys. And I have to say, this has now spilled over to TWiV. We're having a discussion there as well. We're getting lots of emails. And we discussed this last time. And Suzanne wrote, "You're right. I do hear women saying guys, plural to others in a group of women. But I still argue that if I said that guy over there, you'd be more likely to expect to turn and see a man than a woman. So it isn't really neutral. I vote for the spread of y'all." P.S. I catch myself.
Where's she from?
She's from Texas.
Oh, no wonder.
Yeah, they say y'all.
But you can say you all.
You can say you all. Yeah, well, I've been trying
It's
to
not the
purge
same. It's a different word.
P.S. I catch myself at it too. I will also try to be more vigilant. Yes, I've been trying to be vigilant as well. And then we got an email from a colleague of Michele Patschloss. He writes, "Hey gals, although I'll never be accused of being part of the language police, I was taken aback by Vincent and Michael's reaction to Michelle's comment on TWiM 125 regarding the use of guys when referring to people that are involved in computer programming. As you may know, it is thought that there are many small biases throughout the career development of female scientists that discourage them from pursuing specific disciplines, particularly computer science. So it would be worth reconsidering the collective nouns we use to refer to people within science. To bring some data to the conversation, here's a large but admittedly non-scientific survey of 2,300 individuals, which he links to. The survey showed that men are more likely to think that guys is gender neutral when referring to people within a computer science context. Regardless, I think we owe it to our female colleagues and trainees to use the terms that are the most inclusive. As someone that runs a lab with a heavy bioinformatics focus, it is very important to me to maintain a strong balance between men and women, knowing that this is difficult given the poor representation of women in computer science. Both programmers that have worked for me have, in fact, been women. To call them guys would be obviously wrong. Well, kidding aside, I would be unlikely to call them gals as well. Instead, I have been working to cut guys out of my language and go back to my Missouri roots by instead saying y'all. Another vote for y'all. If nothing else, every time I force myself to use this odd slang, I remind myself to check my unconscious biases and to be better. Happy sciencing, Patschloss. Okay, yeah, that's a good point. I agree that these unconscious small biases that we all make every day are not good, so I will try also to get rid of them.
Well, remember the early TWiM that we had with Jo Handelsman about her paper that was in PNAS about the hiring bias, looking at gender neutral names on applications versus gender specific names, and how there was indeed an unconscious bias just looking at the same set of credentials.
Which was, by the way, perpetrated by both men and women.
Both men and women. Yeah. Yes, we both have this unconscious bias, so we both have to commit to squelching it.
Absolutely.
Yeah.
And that's always a good episode to revisit when you begin to have a search committee to, commend them to either take a look at Joe's paper or actually just put TWiM on in the morning.
So here at University of Michigan, we have a group that any search committee, once it's launched, has this group come and give them a primer on unconscious bias and how it can affect letters of recommendation and that. So we're trying to just raise our consciousness and therefore have more equitable hiring procedures here at Michigan.
Yeah, that's good. I like that. All right, this has been TWiM number 127. You can find it at iTunes. You can find it all over the internet using your favorite podcatcher app. It's also on Google Play Music now. That's new for us. And we like getting your questions and comments. You can send them to twim@microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you. And it's graduation weekend. So I'm hooding one of my PhD students, Caitlin Flynn, and then the undergrads graduate on Saturday. So lots of excitement in town this weekend.
And then the campus will be quieter for a while, right?
Quieter, yes.
But not silent, of course. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure. Nice to be back.
It is very good to have you back. Are you going to stay for a while?
Yeah.
No big projects for the summer, right?
Well, the usual.
Well, you're going to be at Microbe in Boston, right?
I am. Yeah, you and I are going to be on a session.
That's right.
Talking about social media microbiology.
That's right. And if we have a TWiM scheduled, and whoever is available of all of you are welcome. I don't know who we're going to have yet, but it's on Friday afternoon. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.